This video is a review for the biology Keystone in Pennsylvania. And this is Anchor 6, genetics. Anchor 6 has a lot to do with comparing Mendelian and non-Mendelian genetics. It also has a lot to do with the central dogma of biology. DNA determines RNA, which determines amino acids, which determines proteins. So DNA to RNA to proteins, and that process that's involved. This anchor also includes scientific thinking, but we covered that in the video for Anchor 1. To understand genetics, you need to understand some significant vocabulary. So take a second, pause the video, match the vocab terms with the definitions. Hopefully you came up with some answers that look something like this. But becoming familiar with this vocabulary is going to be essential for doing well and for understanding the principles of genetics. The law of segregation is one of Mendel's 3 laws. Mendel's law of dominance tells us that the capital G, the gray coat, is dominant to the white coat, the recessive lowercase g. So long as there's a capital letter G, that phenotype will show up. Mendel's other law, segregation, tells us that only one allele can be given from each parent, not both, just one. The law of independent assortment tells us that the gray seed coat trait does not influence other traits. So if we distribute these alleles through the process of meiosis, we get something that looks like this, big G as the gametes. And then as those gametes fertilize, then we have the heterozygous combination, big G, little g. If we were going to try to figure out all the possible combinations between two parents, we can do that using a Punnett square. We put the genotypes from one parent along the side and the genotypes from the other parent along the top. Next, we just move the genotype from the side over and the genotypes from the top down. And when we do, we come up with a combination that looks something like this. Not all types of genetics follows Mendel's rules of inheritance. There are some other explanations and principles to apply for genetics. So take a second and match up some of these terms with some of these definitions. Hopefully you got some answers that look like this. Here's a case of codominance, a red flower crossed with a white flower, and the result is a red and white flower. So we still apply the same rules of segregation to see what all the possible combinations are. And we get a result that looks something like this. All of the offspring are both red and white. If this were incomplete dominance, we could do the exact same process where we cross the red one with the white one, and we would find that all of the resulting flowers are pink. Blood types are a great example of not just codominance, where more than one trait shows up, but also multiple alleles, where there's more than one option for each trait. Here we have a type A parent and a type B parent. The type A parent is heterozygous, and the type B parent is heterozygous. So when we match up those possible alleles, we get a type AB, which is our codominant trait being shown up. We have a BO, which is a type B person, a type A person, and then the O is recessive. So we see multiple alleles, A, B, or O, 3 possibilities, not just 2. Sometimes there can be mutations at the DNA level. In the case of mutations at the DNA level, occasionally a nitrogen base can be removed. So if that guanine is removed, then that type of mutation is a deletion mutation. If we add another nitrogen base, then that's an insertion mutation. Or if we just swap the 2 nitrogen bases, that's a substitution mutation. So these are mutations at the DNA level. There are times where the mutations are on the chromosomal level, where the gene is mutated. So sometimes that segment of DNA is copied, sometimes it's deleted, sometimes it's just moved to another gene. In the first case, we have a deletion mutation. In the second one, it's inverted, it's flipped over. In the third, it's translocation. A segment of the gene has come from another chromosome. And the last one is duplication. We have an extra copy of one of the genes. Here are some DNA mutations. Notice that each word consists of only 3 letters. That shows us the codon, the 3 nitrogen bases that make up a codon. Each of those 3 nitrogen bases in the DNA will be copied by RNA, and they will make an amino acid. So the sentence, the complete sentence, because if you add amino acids together, you get a protein. If we add these words together, we get a sentence. The complete protein or complete sentence reads, "The dog ate the cat." But if we insert another letter, like the letter H in mutation 1, everything after the letter H frame shifts. It shifts over. And everything is, after those first couple words, is nonsense. In a similar way, the dog ate the car, a substitution mutation. We've swapped one letter for another, and we have a completely different meaning. In the third type of mutation, we've removed a letter, a deletion mutation. And as a result, the frame shift has gone back one letter, and the subsequent codons are messed up. Silent mutations are when we have this substitution mutation, but the result is the same amino acid. So there's no problems with making and building that proper protein. A nonsense mutation is a mutation where we get a different amino acid, and as a result, the protein doesn't fold or form correctly. Missense mutations are typically frame shift mutations, where after that mutation, everything after it is messed up. DNA is stored in the nucleus, and it never leaves the nucleus. So messenger RNA makes a copy of the DNA segment that we call a gene. That messenger RNA copies the DNA in the process of transcription. The messenger RNA leaves the nucleus, finds a ribosome where transfer RNA matches up the anticodon on the transfer RNA with the codon of the messenger RNA, and that's how we know the next amino acid to put into the protein chain. The process of building the protein is called translation. Transcription first, then translation. When DNA makes a copy of itself, that's replication. So DNA replicates, copied by messenger RNA, that's transcription, where the messenger RNA is copied by transfer RNA to build proteins, that's translation. Here's a sequence of messenger RNA, and above it we have some transfer RNA. And we need to figure out which amino acid is going to go onto our protein chain next. So what pairs with UAG? Well, you'll notice that because it's RNA, we're substituting the thymine with the uracil. There's no thymine in RNA. There's thymine in DNA. A's pair with T's and C's pair with G's. But in RNA, the thymine is substituted with a uracil. A's pair with U's and U's pair with A's. So AUG pairs with UAC. Well, what's UAC? What amino acid would we find on UAC? To figure that out, we would look at a chart like this. We would start here in the center with the first letter, U. The second letter, we move out one ring, A. And then the third letter, we would move to the final ring, C. So if our strand was UAC, the amino acid would be UAC, and that's threonine. Using this chart, we can figure out what the DNA can code for. In this case, we have a DNA strand AAT. So the process of transcription, we need to convert that to UUA. Now that we have this UUA, the process of translation, what amino acid will we add? So starting here in the middle, U, to the next ring, U, to the final ring, A, we find the amino acid leucine. What if we work backwards? What if we start with the amino acid? Can we figure out what the DNA said, or what's the DNA code? If we start with MET here, we realize the messenger RNA code is AUG. If the messenger RNA code is AUG, then what's the DNA code? TAC. So now that I know that DNA strand is TAC, we can translate it and then transcribe it to get the amino acid. There are some significant differences between DNA and RNA. Remember that DNA is double helix, double-stranded. DNA also has the sugar deoxyribose, and it has a nitrogen-based thymine. Whereas RNA is single-stranded. It has the sugar ribose, and instead of thymine, uracil. So make sure if you're looking at transcription and translation, the RNA strand has the thymine substituted with uracil. I hope that helps you with Anchor 6 genetics.